Good day everyone, I am Christine Joy D. Mallorca. Today we'll be discussing Trypanozoma brucei gambiense. Today's lecture will have a deep dive about the distribution of Trypanozoma brucei gambiense, their habitat, morphology, life cycle, pathogenesis and clinical features, immunity, diagnosis, treatment, and finally, the prevention and control. Trypanosoma brucei gambiense is the species responsible for most chronic anthraponotic infections in West and Central Africa. It is primarily found in 24 countries across these regions. It accounts for approximately 97% of reported cases of sleeping sickness and causes a chronic infection that can remain asymptomatic for months or even years. Countries where Trypanosoma brucei gambiense is prevalent include the Democratic Republic of Congo, Guinea, and the Central African Republic, among others. The disease caused by this species, also known as human African trypanosomiasis, poses a significant health challenge in the affected regions. Habitat: Trypanosoma brucei gambiense primarily inhabits humans as the main reservoir, making them the primary host for the parasite. However, this species can also be found in animals such as primates and ungulates. Domestic cattle are considered the most significant animal reservoir for related subspecies Trypanosoma brucei rhodesiense. In humans, Trypanosoma brucei gambiense lives as a parasite in the blood, lymph nodes, spleen, or cerebrospinal fluid. It can cause a chronic infection with individuals remaining asymptomatic for months or even years. The parasite is transmitted through the bite of infected tsetse flies of the Glossina palpalis species. These flies act as vectors for the transmission of the parasite. Morphology: a unicellular parasite that typically has a elongated or spindle-like shape. It measures about 15 to 30 micrometers in length. In the life cycle, during a blood meal on the mammalian host, an infected tsetse fly insects metacyclic trypomastigotes into skin tissue. The parasites enter the lymphatic system and pass into bloodstream. Inside the host, they transform into bloodstream trypomastigotes, are carried to other sites throughout the body, reach other body fluids, and continue the replication by binary fission. The entire life cycle of African trypanosomes is represented by extracellular stages. The tsetse fly becomes infected with blood meal on an infected mammalian host. In the fly's midgut, the parasites transform into procyclic trypomastigotes, multiply by binary fission, leave the midgut and transform into epimastigotes. The epimastigotes reach the fly's salivary glands and continue multiplication by binary fission. Pathogenesis and clinical features. Infection with Trypanosoma brucei gambiense occurs in two stages. The first stage is characterized by the development of a trypanosomal chancre at the site of inoculation within days of being bitten by an infected tsetse fly. This stage may sometimes be preceded by the development of a trypanosomal chancre. The second stage involves the invasion of the central nervous system, leading to neurological symptoms. The clinical features of Trypanosoma brucei gambiense infection include fever, headaches, joint pain, and itching. As the infection progresses to the second stage, symptoms such as sleep disturbance, confusion, personality changes, and neurological abnormalities including tremors and paralysis may occur. There are 4 types of immunity in Trypanosoma brucei gambiense. The first one is macrophages. These immune cells are responsible for engulfing and destroying pathogens including Trypanosoma brucei gambiense. Second, T cells. They can directly kill infected cells and help regulate the overall immune response. Third, antibodies produced by B cells. Antibodies can recognize and neutralize parasites, marking them for destruction by other immune system. And lastly, cytokines. These signaling molecules help regulate the immune response and can influence the activity of different immune cells to fight against the parasite. Diagnosis. Samples like blood, CSF, and lymph node aspirate. Microscopy, gem-sustained blood smears will show the trypomastigotes. Highly specific serological test and culture medium. Treatment. Early stage of African trypanosomiasis is treated with soramine or pentamidine. Late stages of African trypanosomiasis, when CNS is involved, is treated with Milarosoprol. American trypanosomiasis is treated with Nifortimox. For prevention and control, we can do vector control by using insecticides, protective clothing, and regulation and treatment of reservoir host. And that's it for today's video. I hope you learned something.